Welcome everybody to today's iCubed. My name is Jessica Rodriguez. I am the Industry Engagement and Outreach Officer for the Data Science Institute. Thank you for joining us today. iCubed stands for Institute, Industry, and Innovation. These seminars connect our industry affiliates to our students in the larger Columbia community to explore topics at the forefront of industry. We invite data scientists, engineers, or researchers from our affiliates to give a talk about a recent project or a case study. During these educational events, we encourage attendees to ask questions via the chat or Q&A feature. Today, we have a data scientist from Novartis joining us. Today's talk will be JAEGER, Hunting for Antimalarials with Generative Chemistry, presented by William Godinez, who is an Associate Director and Senior Principal Data Scientist at Novartis. Thank you, and welcome, William.
Thank you, Jessica. Thank you for the kind introduction and the opportunity to present and share our work at this venue. We're happy to be doing so. So like Jessica said, my name is William Godinez. I'm a Senior Principal Data Scientist and Associate Director at the Chemical Biology and Therapeutics Department of the Novartis Institutes for Biomedical Research, NIBR. And today I will be talking how we are using neural networks to design novel antimalarials. First of all, I want to say many thanks to the many people who contributed to this work. This is the result of some fantastic team effort, and so I just want to express my gratitude to everybody on this slide, but especially the co-authors of this work, which are highlighted in bold. So by way of introduction, let me talk about NIBR. NIBR is the research arm of Novartis, which is one of the largest pharmaceutical companies in the world. NIBR has offices in Basel, Switzerland, Cambridge, Massachusetts, as well as Emeryville, California, in the San Francisco Bay Area. And it is in the California office that our data science team is located, and where most of the work that you will be hearing about today is carried out. And so that's that picture of our building where we're currently located. So let me give you some context about our work at Novartis. Our purpose is to improve human health by discovering and developing therapeutics to address some of the most pressing needs in human health. Most of these therapeutics take the form of a chemical substance, of a drug. The process of drug discovery is a long one, often spanning several years, up to 10 years, 15 years is common. And this slide shows an overview of this process. So to start with, we need a detailed understanding of the biology of disease, and we need to identify a potential biological target within either cell or a certain pathogen that we want to be targeting. Once that biology is understood, once that target is determined, we can start developing and discovering new compounds, new chemical matter, aim at modulating that particular biological target of interest. And once we have a promising drug candidate, we can move further into developing that drug or that compound into an actual medicine that we can use safely and efficaciously in humans. It is at that intermediate stage of molecule sort of discovery, molecule optimization, where our work with JAEGER comes in. At this stage, especially as we move from what we call a lead compound into a candidate, a drug candidate. Many of our drug discovery teams, many of our colleagues in the chemistry departments are often confronted with the task of designing, synthesizing, which is basically creating the compound, and developing novel chemical matter. So we need to discover new compounds. So that's the state at which our work is making an impact. So let me tell you how we're currently approaching that task of designing novel molecules. This is the sort of traditional paradigm where our chemists would make modifications to a molecule. They would synthesize that molecule. They would test that molecule in the lab. And through those experiments, they would measure various properties of that molecule. So for instance, they would measure how potent that molecule is, how soluble that molecule is, how permeable. Also need to measure some toxicity issues. And so by sort of making modifications to the molecule, you would eventually find a compound that would sort of meet the various requirements that we have for those compounds. So we often want very potent molecules, the potency being somewhat related to the dosage that is needed for that compound. We also, for safety reasons, we don't want that molecule to be toxic. So this various sort of permutations of those molecules are time consuming, are very resource intensive. And so we're always looking into ways as to how we could improve or speed up that process. So there's a sort of different paradigm to approach this, what we call an inverse design for drug discovery. And so instead of starting with the molecular matter, we start with actually prescribing the sort of profile or the criteria that we want to meet with our molecule. So we can start saying, okay, we want very potent compounds, we want very safe compounds, we want compounds that will target specifically the target of interest. We don't want the compound to be hitting multiple targets, and that's what selectivity is referring to. And you can sort of prescribe all of these criteria and then give it to a computer, give it to an algorithm, and let the algorithm figure out what would be the best sort of molecule or set of molecules that would meet those prescribed criteria. And so this idea is not necessarily a new one. This was explored earlier, perhaps in the 80s and the 90s. This was the so-called de novo design algorithms that would basically from scratch build a molecule that would essentially do that. That would match any sort of prescribed criteria for potency or and so on. The problem being back then that many of the molecules that those algorithms came up with were hard for the chemists to actually synthesize in the lab. So it may not have had as much success or it's not as a widespread technology as it probably could have been. But now with the advent of deep neural networks, and I'm sure many of you in the audience might be familiar with the results that you can do with those methods, for instance, with images, where you can sort of dream up a new sort of landscape or dream up a new face of a person. The sort of analogous idea that we could use that in chemistry is, could we let the neural network instead dream up a new molecule that would match our sort of set of criteria? And that's the whole idea behind what we call generative chemistry. So generative chemistry, in a nutshell, is the use of deep neural networks and mathematical optimization for the targeted generation of new molecules. And here the key word is targeted. We're not going to perform enumeration of all possible molecules, as you can imagine, all the possible combinations of atoms and bonds is a quite large space from a mathematical standpoint. So it just, and there's people that are actually taking that approach, and that's fine, but the generative chemistry flavor is more of a targeted sort of approach towards the problem of molecule generation. Any generative chemistry method is composed mostly of three components. And here you can see the three components. I have listed them: molecular representation, a neural network architecture, as well as a mathematical optimization strategy that is used for finding the new molecules. So what I mean with molecular representation is the question as to how do we represent a chemical compound? How do we represent a molecule within a computer? And more specifically, how do we represent that within within a neural network? So most of the times, a sort of a very common approach to represent molecules is through a string-based representation. So basically, a set of characters that represents the structure of the molecule. Basically, it's atoms and its bonds that sort of conform that molecule. And this is what we call SMILE strings. That's the first item in that list for molecular representation. Give my pointer. Then we also have what we call fingerprints. These are vectors where each vector in each where each element of that vector sort of represents what kind of substructure or what kind of atom we might have within the molecule. The advantage of the fingerprints is that we have as a vector of a constant size that sort of is the same size for all the molecules. As you know, some of the molecules might be smaller, might have fewer atoms, might have fewer bonds, and some other molecules might be pretty large, might have pretty long many atoms and many bonds. And so the question is, how do you represent that structure with a data structure that has a constant size? And so the fingerprints address that issue. So that's one way of representing the molecules within a neural network. Another way is to actually use the molecular graph. As you may know, a molecule is a set of atoms that are connected by bonds, and you can represent that through a graph. Another sort of popular way to represent the molecules is actually a 3D structure. As you know, actually the molecules have a 3D structure, and so they can be represented geometrically. So you could actually embed that 3D structure into a discretized grid that you could also consider as voxels. So that's just another way of representing molecules within a neural network. The representation will dictate strongly what kind of neural architecture you can use. So for instance, if you have SMILES, which is a set of characters, set of strings, you can consider that as a sequence of characters. So you could use some sequential method for that kind of representation. So you use some type of recurrent neural network like an LSTM or a group. If you have, for instance, a molecular graph, you can use variational auto-encoders. You can use also generative adversarial networks and so on. That's just a neural architecture. You also have to consider, how are you going to use that neural architecture to generate new molecules? How are you going to harness the power of the neural network? And that's where these optimization strategies come in. So very commonly, you would use some gradient descent within the latent space of for instance, a variational auto-encoder. But if you have something like a recurrent neural network, you can also employ Monte Carlo tree search, where you sort of explore in a relatively stochastic manner the space of molecules. So the different combinations of the different sort of components will give you different methods, and there's a lot of methods in the literature. And here in this table, I show you in the left the many. It's a literature review of some recent work in generative chemistry, and it shows you sort of the breadth of work that is being done in this field. So it's a pretty active field. Primarily, however, most of the papers only propose methods. They don't go all the way into the lab where they test some of the molecules that have been proposed. And that's why some of these papers that I have here on the right are a little bit more prominent, because they actually managed to sort of bridge that gap, sort of going from the method all the way to an actual molecule that actually works in the lab. But they're still very sort of rare and not very common in the literature. So that's where our work comes in, where we develop JAEGER, which stands for JT-VAE Generative Modeling. And JT-VAE stands for the Junction-Tree Variational Auto-Encoder, which is a particular architecture that we're using. As a side note, Jaeger means hunter in German, and we at Novartis, we're in the business of hunting for drugs. So we're drug hunters, since the name is sort of an allegory towards our activities. But Jaeger was developed internally here at Novartis in order to bridge again that gap between the models and to bring and leverage our data to generate new molecules that are actually active in the lab. So we have proposed efficient numerical search strategies, and I will speak about them in a second, to sort of generate these novel molecules. And we are, as far as we know, as far as we can tell, we are the first team to successfully design inhibitors for malaria that actually have been experimentally proven in the lab to work. The workflow on the top of the slide is just shows a little bit more sort of pragmatically how we employ Jaeger within Novartis. At Novartis, we have multiple assays, so multiple experimental protocols where we study a specific biology. So for instance, we are interested in a certain target within a certain cell line. So for instance, within a cancer cell line, for example. And so we measure the specific activity of compounds within that cell line. Are the compounds sort of, let's say, inhibiting a particular target and sort of, let's say, killing the cells? That's the activity that we have, and we have thousands of those assays. We have also many biologists working with us and internally at Novartis. And so what we do is we take the data from one particular assay. So basically, that's a list of compounds with at potency, what we call AC50, and I will explain in a second what is AC50. And we build a model. We build a Jaeger model, which is basically an Auto-Encoder. Then we generate the molecules, and then we pass those generated molecules through other, or what we call orthogonal in silico validation tools that at Novartis have been developed over several years, and that are proven to be very accurate when sort of predicting the activity of those molecules in our assays. And then once we sort of filter those molecules with this orthogonal validation schemes, we can proceed to the lab, and together with the chemists and the biologists, decide whether we can actually use and sort of validate the proposals of our method. So let me talk about Jaeger and the different components within this workflow. Let me explain to you how generative chemistry works with variational auto-encoders. That's a particular flavor underlying Jaeger. So we have a set of molecules, a set of compounds, of existing molecules that we have actually tested in the lab, that we have actually synthesized. And we train an encoder. This is just a neural network that maps those structures into numbers. It's just a vector, what we call a latent representation of those of a molecule. That latent representation, or that vector, usually has multiple dimensions, up to 56 dimensions, 128 dimensions. But just for illustration purposes, you could think of it as a 2D space, a 2D map. So each molecule in the collection sort of maps to a particular coordinate point within that map. Any coordinate point is also can be decoded again through another neural network that we train into a molecule, into the same molecule, right? And that's the concept of variational Auto-Encoder. Within this latent space, you can start building what we call activity models, where basically you map any point within this latent space, any coordinate point, and predict its activity. You can train such a model because you have measured the molecules in the assay, so you have those activity values, those potency values. And as you do that, you will start populating this space, and you will start sort of seeing what's the landscape of activity of potency within this latent space. And as you if you sort of overlay a surface, interpolate a surface over all the data points, you will start seeing, oh, there's like a sweet spot there. So the whole idea is to try to move the molecules towards a sweet spot, towards a local minima or maxima, depending which direction you're trying to optimize. And then once you're in a different sort of coordinate point, you will have a different sort of latent vector representation that you can feed to the deep neural network, and you will come up with some new molecule that could be potentially an improved molecule compared to what we have seen before. All right. If there's any questions, please feel free to interrupt me at any time. Right. So, like I said, the whole idea is to optimize mathematically in this latent space to find the best compounds. That's where the mathematical optimization comes in, and where we're basically going to transverse this latent space. The particular flavor of a particular architecture that we're using with JAEGER is the Junction-Tree Variational Auto-Encoder JT-VAE. This was developed by a lab in MIT. And this model represents a molecule through two information pathways. The first pathway sort of represents a molecule through a junction tree, and I will explain a little bit what a junction tree is, but in essence, it's just a coarse representation of the atom clusters within the molecule. The second pathway is building a representation directly on the molecular graph, so the atoms and bonds within the molecule. Each pathway has its own sort of encoder, so you have actually two latent vectors, as opposed to the earlier slide where you only had one single vector. Here, the molecule is represented through two latent vectors. The latent vectors sort of are concatenated, and we build a sort of activity model that sort of produces the potency of the molecules that will be used for optimization later on. And also those vectors serve as input for another decoder that reconstructs the molecule. For JAEGER, we made some modifications to the activity predictor, mostly some hyperparameter changes compared to the original publication, and also some GPU memory optimization. All right, so why are we using the JT-VAE? What's the rationale behind it? So, like I said, the molecule is constituted by various atoms and bonds, and this the sort of bonds sort of interconnect different atoms, and you could sort of cluster those different substructures, and you would obtain what we call a cluster graph. Then using a minimum spanning tree algorithm, you can obtain a junction tree. And sort of the beauty of this approach is that you will have a representation of clusters that overlap with each other. And the overlap is important because as you reconstruct the molecules, you will be able to sort of match the different constituents, the different clusters that are compatible with each other. That is very helpful towards generating 100% valid structures, which is a very key aspect of this method as well. So as I was alluding, when doing reconstruction, we're not going to reconstruct atom by atom, as that leads you into sort of intermediate chemistry or chemical structure that might not necessarily be a valid structure or a stable structure. And so, for instance, if you sort of, for some reason, the optimization doesn't close the ring, for example, that we see here, that might not necessarily be a compound that might be amenable for synthesis, and the chemist might just not like that. So when you're using the junction tree, you will generate the structures sort of cluster by cluster or structure by structure, and so that will give you compounds that will be chemically valid at each reconstruction stage. And so here I have the table that just sort of compares the validity of the molecules that are produced by JT-VAE, and so that's 100%, and that compares so much better than what was previously described prior to the publication of JT-VAE. So that's one of the reasons why we chose this method as our architecture of choice. So maybe getting a little bit more technical, just to give you some of the of the details that we have to deal with when we're training a neural network like this. We need to sort of optimize various loss functions. And here I have the loss function equation for this particular architecture. So we are optimizing first a tree reconstruction loss. That's just telling you or measuring how well is the method reconstructing the actual junction tree, so the various clusters. Then there's a loss related to the graph assembly. So that's actually, again, how well is the method reconstructing the different graphs, predicting the different subgraphs at each tree node. Then we have variational loss, and that's just, we apply this loss to sort of smooth out the latent space to maintain a sort of a Gaussian prior within the latent representations, so that they do not collapse into a single sort of area within the latent space. Then we have an activity loss, that's for the prediction of potency, and we have a stereochemistry loss, which is related to the fact that molecules are actually, like I said, 3D entities, and so we have a predictor within this method that sort of tries to predict the right conformation in 3D. And so we train this in a two-stage fashion, where sort of we turn off the variational loss. The beta is sort of set at 0 at the beginning, and so that we let the method learn how to reconstruct the trees and the graphs, and then we apply this sort of variational loss later on in a second training stage. All right, that's so much for the Auto-Encoder, for the neural network architecture. Like I said, there's still the component within a generative chemistry approach as to how do we explore that latent space. And there are various approaches. So what we have here in this representation is, again, a 2D projection of the high dimensional latent space. And so in this case, I used PCA. And I overlaid this, the surface that you see here is related to the PAC50. PAC50 being the negative logarithm of the AC50 value in molar concentration. AC50 being, again, is the sort of, it's just a measure of how potent the molecule is. So here the values range from 5.5 to 8.5, with 8.5 being the more potent molecules and 5.5 being the sort of less potent molecules. And so here, for instance, we have a starting molecule, and I represent that with this red dot. And so the question is, how do we move around this surface, which is actually rather fragmented? How do we move around to sort of find a new optimal molecules? How do we move to these sweet spots? And so you can think of it using, for instance, gradient ascent, where you can perform gradient ascent on the function predicting these PAC50 values. But that is, the gradient is actually rather noisy within these areas. The surface has moved out actually the various values. So it gets stuck easily on some local minima. So we don't like that approach too much. You could also smooth out the surface with Gaussian processes. Still you could perform, once you smooth out the surface with a Gaussian process, you can still perform gradient ascent, but that's still sort of subject to like the non-convexity of the activity surface. As you can see, there are multiple sort of optima here within the surface. So it's kind of hard to do gradient ascent or descent. We prefer to sort of hedge our bets and actually perform searches in different directions. The gradient is a single direction, but you could think of exploring multiple directions. So that's actually our method of choice. And we have also explored sequential Monte Carlo methods that sort of adopt a more stochastic method. I will explain briefly this neighborhood search methods in the next couple of slides. So basically what we do is we define a Gaussian around this sort of a starting point, the red dot, and the Gaussian is derived, is sort of defined here. It has parameterized by a mean vector and a covariance matrix. The mean vector being actually the red dot, and the covariance matrix is a scaled version of the covariance matrix of all the training samples. And so you can just use sample or some stochastic samples around that, and that will give you multiple directions along which you can explore the latent space, and you could sort of start converging onto some of the local minima. This is a sort of smart approach, but it's actually relatively time consuming because of the multiple dimensions of the latent space. We have maybe 56 or 128 dimensions. Then you need a lot of samples to actually get a good approximation of the, or a good sort of search efficiency of the latent space. So what we're doing is actually using the eigenvectors of that covariance matrix, that we defined for the Gaussian, and actually just search along those main sort of axes. And so I have here the equation where the eigenvectors are sort of scaled and just added to the starting point. And that actually has proven to be pretty efficient within this high dimensional space. All right, let me move to some results. We applied JAEGER to malaria. This has been a work with the Novartis Institute for Tropical Diseases, which is also co-located here in Emeryville. Malaria causes almost 500,000 deaths annually, mainly in children. And the parasite is developing resistance to most of the therapies that are available. And so that's why NITD is heavily invested into that disease. And so the idea from one of our colleagues, Armand Guigon, was to, hey, let's use JAEGER and design some novel antimalarials. So the data that we use to train the data set comes from an internal malaria Plasmodium falciparum assay, where actually here I have an image of the mosquitoes that are actually used to extract the parasite. So for that data set, we removed the compounds with more than 50 atoms. So compounds that are relatively large. We did not include them in the method because the method has some issues with reconstructing larger compounds. Remove some of the compounds that had AC50 values or potency values outside of the dose range. And so we ended up with 21,000 molecules with measured potency values, AC50 values. Here are some of the architectural details that we use for the JT-VAE. The number of the dimension of the helium layers is 420. The dimension for the latent spaces, so 28 dimensions for the tree latent space, 28 dimensions for the graph latent space. Number of message passing rounds within the graph convolutional networks is 7, and some additional details regarding the training that I have here. So it takes about 135 hours to train this model. It's almost a week, a bit more than a week on a sort of a little bit older GPU that we use for that. Here, just for fun, I show you the loss traces. Maybe the most prominent one is the variational loss. As I said, the beta is sort of disabled at the beginning, at the first stage of training. Then we sort of turn it on, and then you can see it starts converting. Couple of validation points for the model. So we try to measure through cross-validation. How well are we reconstructing the trees? So this is the error value, it's 6%. How well are we reconstructing the graph? Also again, an error value. We also measure how well we're predicting the potency, which is an actual scalar value, the PAC50. So we measure the mean squared error as well as the correlation, the squared correlation coefficient. Also, just for understanding of what's going on in the latent space, what is a network learning? I think that's a question that often comes up is, we perform first PCA on the latent space, and on the 3D space, we see that I'm plotting here the explained variance, the proportion. So it sort of goes all the way from 0 to 1 as we sort of add it up. It's a cumulative function, and then as a function of the index, of the principal component index. So you can see that we reach about 90% variance with about 20 to 223 principal components. So that tells you about how efficient is the model using all the available dimensions, because we have 28 dimensions, but maybe only 23 are sort of effective in a way. For the graph space, it's actually a very different picture. Actually, already with one or two latent space principal components, we are already covering most of the variation within the data. We see how the principal components also, we check how do they correlate with actual some of the chemical properties of the compounds. So for instance, TPSA being the topological polar surface area. And we see there's a relatively not so strong correlation, but it's not necessarily lower either. So there's correlation with some of the principal components, as well as, for instance, the synthetic accessibility score. There's also some correlation with a principal component within the graph space. And this diagram on the left is for the tree space. So what this tells you is that if you sort of explore linearly along the principal components, which is what we're more or less doing, you would ensure that you get a little bit of diversity in terms of the different compounds that we're generating. All right, so we applied this, and we used a deterministic neighborhood search that I described earlier. We used 3 starting points, and we generated 4.2 thousand latent space samples per seed. And then we sort of set a criterion for filtering the molecules of PSA 50 of 8. And so once we sort of filtered everything, we had 282 unique and novel virtual molecules matching the criterion. And so here I just plot the histogram of values comparing the 282 molecules to the training set. So in the training set is shown with the orange histograms, and the generated set is shown with the blue histograms. And you can see that the sort of overlap between the two histograms are relatively good. Here I'm showing the properties of the molecules that we generated. So we're looking at the molecular weight, which tells you a little bit how large the molecules are. The calculated log P being the octanol water partition coefficient, which basically measures how greasy in a way the molecules are. This is sort of an important parameter, because as you can imagine, if you have ever cooked and you know how things that are greasy sort of stick to everything. So you don't want that necessarily. You don't want the molecules to be too greasy. And so on, we have other parameters that we sort of monitor to make sure that the molecules are sort of making sense. And so in general, what we see is that the molecules generated by JAEGER are sort of very realistic when we sort of compare it to the training set, which is actually existing data. All right, so we've further filtered the molecules with a method known as Profile-QSAR. Profile-QSAR is a huge multitask machine learning method that is very accurate towards predicting activity within assays. So with Profile-QSAR, we selected 4 molecules, and out of the 4 molecules, we managed to synthesize 2 molecules. And here are the properties of these 2 molecules. Here are the structures of these 2 molecules. And here are the predicted potency values. This is a potency value predicted by JAEGER. This is actually pretty high. A pQSAR is a little bit, has a more conservative prediction. And the molecules are accessible in 6 synthetic steps, so it means it's just not that difficult to synthesize these molecules. The question that we often get is, how novel are these compounds? And so we measured the similarity of these compounds to the training set through what we call a tiny model similarity. That's a measure that is used to compare compounds that ranges from 0 to 1. So 1 being very similar or the same compound, and 0 being not similar. So we can see that most of the compounds in the training set are not similar to the compounds that we generated. We measure similarity to the nearest neighbor in the training set. It's 0.65, 0.67. It's not too similar, but not exactly the same. Similarity to the seed molecule. So we did diverge from the seed molecule that was used. And again, also we checked the Novartis archive, which has about 5 million compounds. And again, we just don't find any molecules that are similar. So that sort of tells you that these molecules are pretty novel. So finally, we went to the lab. That was pretty exciting to actually test these molecules in the lab. And will they like inhibit malaria? That was the question. And so here I show the activity of these molecules in 3 assays, the phenotypic assay being measuring just actually where they're actually inhibiting malaria. The biochemical assay is actually measuring whether the compounds are hitting a particular target within the pathogen, within the parasite. And then we also measure cytotoxicity just to make sure that we're not killing cells arbitrarily, because we don't want to kill the human cells, we want to kill the parasite. So overall, the values were pretty good, very potent in the phenotypic assay, but it was still potent, but very potent in the biochemical assay. So that was pretty encouraging. Those were pretty encouraging results. So we did a second round, retrained the model because of chronological reasons, and we just actually the random neighborhood search in this case. After some sort of filtering through the criteria of potency and also regarding some of the structures that needed to be preserved within the structure, we selected 7 for synthesis and validation in the lab. And so those compounds I cannot show you because of proprietary reasons, but some of the compounds were proposing designs that the team didn't think were going to be active. So when we actually tested those designs in the lab, and they were actually proved to be very potent, especially this one was very potent, the team was pretty surprised and unexpectedly sort of happy about that result, because it was just something that you actually want with this method to sort of start generating ideas that our chemists might not have necessarily predicted that would have been active. There was one particular compound that proved to be very active in the phenotypic assay, very active in the biochemical assay, and that was potential what we call front runner, which is a very sort of promising candidate. It was sent to further evaluation, so it just turned out that it didn't match the physical chemical requirements that the team was looking for, but it just goes to show that JAEGER is sort of generating molecules that are inspiring the chemists. To be fair, this compound that I'm showing here was also found out at about the same time by the chemistry team. So it was just a sort of, in a way, funny coincidence, but at the same time sort of validating the designs by JAEGER. Right, so today I just talked about what we're doing with malaria. There's various other projects within Novartis that where we're testing this methodology. We have sort of synthesized about 9 compounds for malaria and a number of other compounds for the other projects. And many of them have been active. So far, JAEGER has been fairly successful. So this brings me to the end of the presentation. I have shown you JAEGER, which is a generative chemistry approach that couples JT-VAE with efficient latent space exploration strategies. JAEGER generates 100% valid molecules with proven potential in the lab, and JAEGER is starting to augment the chemical reason of our internal teams. A sort of outlook, we want to introduce additional information into the generative process. There's so much data that we have here in Novartis, and so there's so many ideas into how we can improve these models. Also, we're thinking about more sort of in the area of the deep neural network, how can we make it better? And also, sometimes there are some assays where we don't have the many thousands of molecules as we had with the malaria assays. So that's also another issue where we are operating in a sort of data reduced regime. I just want to highlight that there's opportunities in our team in the Bay Area. So if you're interested in working with us, please go to the Novartis careers website and you'll find opportunities there. So thank you very much for your attention. And if you have any questions, I'll gladly answer them.
Wonderful. Thank you so much for the very interesting talk. We actually do have a question. I'm going to go ahead and read it out loud for us. Does the use of machine learning models in the optimization of the drug formulation shorten the discovery stage? If not, why? When training the models, are you using quantum computing to address GPU limitations?
Right. I will address the first one. So we're not training the model to improve the formulation step. We're a little bit earlier. We're a bit before that step of formulation. We're actually just looking for potency, to improve the potency of the molecules. And I guess we haven't really measured whether it has improved the time towards finding molecules, whether using the model would be faster than just doing what we have been doing so far. I think that would be an interesting experiment, but it would mean that that we would have to be totally independent from our chemistry teams, which is not necessarily the most realistic scenario here, and not necessarily the most optimal scenario, because it does take a lot of interaction with the chemists to sort of fine tune this type of models. So I don't think the model is meant to sort of replace or the chemists. I think it's more to augment the capabilities of our chemists. And regarding the quantum computing question, no, we're using standard regular GPUs. I think there is interest towards looking into that area of using quantum computing and speeding up the computation time. That is certainly something that would be welcome to have models that train much more quickly.
Awesome, thank you so much. So I will take the opportunity to ask a few questions, since you are there are openings on your team, and our students are often interested in the type of information that they can garner from our affiliates during these sessions. So what would make a strong candidate for someone to join your team?
That's a good question. I think we're looking for independent thinkers, somebody that has their own ideas with regards to the data science algorithms, with regards to how can we use data science towards driving impact in the lab, driving impact with the different projects that we have at Novartis. So I think that's one key element that we're looking for. I think, of course, maybe it goes without saying, but we look for someone with good programming skills, especially Python or R. Those are the main two programming languages that we look at. And finally, we're looking for someone with very good communication skills. As you know, here at Novartis, we collaborate internally with chemists, with biologists. They have different backgrounds and different different ideas about how to approach the work, and so you have to be able to communicate your own ideas and your own results in a sort of way that is amenable to them as well. So that's also a very key aspect that we're looking for.
Absolutely. You answered my second most frequent question, which is what are the definitive skills? Also, the Python and R. That's really good to know. Thank you for our students. Another question that we often get is, what does a regular day look like for you? Would you be able to walk us through that?
Yeah. My day usually starts with checking my emails. I get a number of emails that sometimes, especially because we're a global organization, so we have offices in Basel, offices in Cambridge. So as you can imagine, in the middle of the night, I might get an email from someone in Basel, so I don't want them to keep them waiting for too long. So I try to reply. So I have a look at my emails, and it depends. So for instance, if I have to do a presentation, I have to work on the slides. If I am working on training a method, then I open up my editor, text code editor. I use Emacs, if anybody knows that. And I don't know, I start sort of tweaking my algorithm, my data. I have, like I said, we have meetings where we share ideas, where we present results. There's seminars where we learn about other people's work, also in the other sites. There's group meetings internally. Our data science team meets about once a week. So it's mixed, I would say. It's not like I sit in front of the computer and code the whole day. I don't think that happens that often. Some days maybe, but many times I do have to communicate with people, which is I enjoy too.
Awesome, thank you. We do have a question that came in from our audience. How was the number of latent space dimensions determined during hyperparameter tuning? Was the analysis to view the variance explained per principal components used for that?
No, we took the default parameters. The issue with this model, or one of the issues, is the computation time. So, and also we don't have that many GPUs that we could use for hyperparameter optimization. So we just took the default parameters, and that seemed to work well. The only hyperparameter optimization that we did was for the number of rounds in the graph convolutional neural network. That one is a bit of a module that you can sort of extract independently from the model and use it basically as an independent module, and you can perform optimization there. For the latent representation within the JT-VAE, I don't think you can do it independently, so we didn't do that part. Yeah.
Great, thank you. Another question, is the position that you posted or you're talking about only available to PhD candidates, or are master students eligible too?
Most of them are for PhDs, but it's not a requirement. I think someone with a master's and a bit of experience would be, of course, very welcome to apply. I think at the end of the day, we are judging for the achievements, but also the potential that that person might have.
Right. Always apply, students. That's what you're why not?
Yes.
If it's of interest and you have the innovation and interest, go for it. Another question that I often get from our student audience is, what kind of data do you work with on a day-to-day basis?
Yeah, so my background is in computer science and specifically computer vision. So I also work with imaging data.
Mm-hmm.
So that's data coming out from microscopes where we observe the cells and we observe what kind of phenotypic outcome the compounds are using. So what kind of geometric shape do the cells have? Are they like elongated? Are they round? And so we also use deep convolutional neural networks to analyze that type of. The other data that I work on is sequencing data. So these are data that measure the activity of genes within different cell lines. And so we also sort of measure how our compounds are modulating that signature, what we call a signature of genes. And so again, we have different methods, but also we are our team focuses on developing and applying deep learning methods towards that type of data. And like I said, like you saw today, we also work with molecular data, the compound data. Mm-hmm. That's the third sort of main data type that we work on.
Awesome. And you mentioned a little bit about your background, but it's always wonderful to learn about how did you get to this place? How did you get to a career in data science?
Right. My background is in computer science. And when doing my master's thesis, I studied in Germany, by the way. Mm-hmm. And for my master's thesis, I did it with a professor at the German Cancer Research Center. So that's already, I was already working back then in sort of the biomedical space. And after my master's, I did a PhD with the same professor in computer vision applied to biological images. So I was developing tracking algorithms for virus particles, so HIV or hepatitis. So you can see the viruses within the images. And then I moved, after my PhD, I moved to Novartis in Basel, Switzerland. They had an opening, a postdoc opening for someone with my profile. Actually, the task was to develop deep learning approaches for high content cellular images. And after that, I found an opportunity here in California, where I'm located right now. And that's more or less my trajectory. So yeah, my background is computational, but I've been always a bit exposed or within that biomedical area.
And interest. Wonderful. Thank you so much. So I really want to thank you for today's talk. It's really informative and it was so interesting, and for telling us so much about Novartis and the opportunities that are there for our students. And I thank you for your time today, William, and we hope you'll be back and you'll engage with us again in the future.
Thank you very much.